So the Baltimore classification of animal virus is probably the most used classification system that we have. And what you can see is that there's 7 different types of animal viruses. Some are single-stranded RNA, some are double-stranded DNA, but whether you're DNA or RNA, by hook or by crook, you're going to make messenger RNAs that are going to be translated into proteins that the virus can use to make more of itself. And so what we're going to focus on is HIV and the coronaviruses. So what you see over here in number 6 is that HIV is a plus single-stranded RNA that has a DNA-RNA hybrid that ultimately will go to double-stranded DNA that we know can integrate into the chromosome as a lysogen, and then ultimately it can be expressed into RNA. On the other hand, coronaviruses, they are plus-stranded single-stranded RNAs, and they do not have a DNA intermediate, so they're always going to be in the lytic phase. So lytic or lysogenic. Crick Central dogma was really important in the 1960s. Crick came up with the concept that DNA was turned into RNA, and then the RNA could be translated into protein. But the discovery of retroviruses threw the world on its head. For example, the discovery by Baltimore and Temin of reverse transcriptase showed that RNA can be converted into DNA by an enzyme known as reverse transcriptase. And what do you see with reverse transcriptase over here? Well, what you see is it has the capacity to take RNA, use it as a template to make a DNA-RNA heteroduplex. So then reverse transcriptase acts as an RNA-dependent DNA polymerase, which is completely opposite of what humans have because we have DNA-dependent DNA polymerases. Now, RT, or reverse transcriptase, isn't done with that trick. Rather, it has another one. It could actually chew away the initial RNA strand, which is shown in black here. So then reverse transcriptase can chew away the RNA, leave the DNA, and use that DNA as a template to make a double-stranded DNA molecule, showing us that RT has not only an RNase activity, but also a DNA-dependent DNA polymerase activity, which is pretty remarkable. We're going to see RT coming up pretty soon because it's important in molecular biology. So what about retrovirus structures? So what you see here is a schematic for HIV, and what you notice is that here's the genome, but you also notice that there's enzymes such as RT. So the actual DNA is in the capsid, but you also have to package reverse transcriptase enzyme itself along with a host of other proteins that we won't go into. But what we also notice, besides the fact that RT is packaged as an enzyme into the capsid, is that the outer lipid envelope here is derived from the host CD4 T cell, because we see host proteins such as MHC, or the major histocompatibility complex, is actually part of the virion structure, along with things like GP120, which is the receptor that HIV uses to get into the cell. So the knowledge of reverse transcriptase and HIV led us into developing antiviral chain terminators. And how do they work? Well, the simplest way to think about them is at the 3' hydroxyl group. This serves as a nucleophile to attack an incoming base at the alpha phosphate position. And that's how you form your new phospho and hydride bond. So you need to have a free hydroxyl group at that position in order to make another growing chain of a virus, or any nucleic acid for that matter. So what about developing drugs that actually don't have that 3' hydroxyl, but rather have other groups, as we see here, the azo group with azithromycin, or AZT, which was the first antiretroviral therapy approved by the FDA for HIV. And you may have seen the Dallas Buyers Club with Matthew McConaughey. It's actually a true story about a rodeo cowboy from Dallas who actually was trafficking HIV drug AZT back in the day when it was pretty difficult to get. It's an awesome movie. So now let's look at coronavirus as an example of positive single-stranded RNA viruses. And here's an example of coronavirus. There's lots of coronaviruses. There's SARS, there's MERS, and the one we're looking at now is SARS-CoV-2, which we believe is derived from SARS in some capacity. So the coronavirus name comes from the fact that it looks like a crown. So here's a graphical representation of a coronavirus. what you see is the spike proteins, and you see the red positive stranded RNA that comes into the cell through the ACE2 receptor, and a positive single-stranded RNA is tantamount to a messenger RNA. It's like a sense strand. So the host ribosome will translate it and make a bunch of proteins, as you see here. And one of the proteins that it translates is an RNA-dependent RNA polymerase, which will use the genome as a template to make more negative strands as a methodology to propagate itself, whereas these other proteins here, the non-structural proteins, as well as proteins that are going to be part of the virion, can actually go through the secretory apparatus, get put into a vesicle along with the genome, and what we do is we exocytose a brand new virion. So what you see is that coronavirus is a positive stranded RNA molecule that's translated. Now, people have come up with remdesivir. Remdesivir is to coronavirus is what AZT is to HIV. It's the first generation FDA approved drug for coronavirus. So what you see here is, by taking a look at it, is that it looks like kind of like a purine, and it's true that the cell actually uses this to make more viral particles, and what happens is that this is going to ultimately inhibit the capacity of the virus to further replicate its genome. So it's absolutely a replication inhibitor. And so we've talked a lot about coronavirus testing. You hear it on the news all the time. You hear the PCR test, you hear the antibody test. So what does that really mean? So let's think about what we know about coronaviruses and apply it to what people talk about in the news about tests. So the PCR test is for active infections. And what you do is you take a swab, perhaps a nasal swab or saliva, and you isolate from those epithelial cells that are infected the actual RNA molecule. But in order to do PCR, you need double-stranded DNA as your template. So what do? You use reverse transcriptase in the molecular biological lab setting. So people use RT as an enzyme in PCR assays. And what we do in this case is the coronavirus will be propagated into heteroduplex of RNA and DNA. Then RT will make the double-stranded DNA, as we discussed earlier. And then what you do is you put PCR primers that are specific for coronavirus to amplify the signal, and that will tell you whether or not your patient is positive or negative for the signal. And of course, you use a positive control. Now, the original CDC tests have been widely mocked for the fact that they were defective, and a lot of people say they were defective because the primers didn't work. They weren't specific for coronavirus, but rather could give false positives, for example, by amplifying other sequences that are considered contaminants. So if you're positive for a PCR in the coronavirus test, that means you are actively infected because the virus is only in the lytic state. Now, on the other hand, we have the antibody test, and how does that work? Well, when you're exposed to the virus, what happens is that you're going to develop an immune response. You're going to have macrophages or other types of cells presenting to T cells, which will ultimately cause B cells to express IgM on their surface. Now, IgM is a relatively weak affinity for the antigen. And what will happen, though, is that some of the B cells will be committed to memory. And when you're exposed for the second time, what happens is that you generate huge proliferative capacity to make more immunoglobulins against that particular antigen. And in this case, the IgG is stronger and it's secreted, and therefore you've developed antibodies against the coronavirus. But just because you have antibodies doesn't mean that you're immune. So having antibodies is necessary